so this was a really fun lab, and basically what we did was we tested whether something is GMO or not. We did plants, and that was fun. So I brought in a bean from my friend's lunch, which was interesting. And so basically we have a non-GMO control food and our test food. And the non-GMO food was, like it said on the package that it wasn't GMO, it was wheat, I believe, or some kind of grain. It was fun. And then we have the Instagene, which basically makes it so that it doesn't bind to itself, and that it binds to the plant and the GMO primers. So what the plant and the GMO primers do is they make it so that you can tell, okay, when we put this into the electrophoresis thing, we can tell, okay, so this is GMO and this is not, because we can tell that it's different cuts. So anyway, basically what we did is we put all of our ingredients into these tubes. We had a very long, painstaking process of grinding the foods so that we can micropipette them. It was a long process. Okay, so we used a water bath again, just so that the enzymes have an opportunity to activate, because they need heat to do that. Okay, so in our tubes we had two tubes that contained each of the foods, and also we had a GM positive plasmid and a GMO positive plasmid. So we would have 2 tubes of the non-GMO DNA. We would have 2 tubes of our test DNA, and then we would have 2 tubes of the GMO positive plasmid DNA. Okay, and so we have the plant primer, which was green, and then let's just say this is red. And then we have the GMO primer, which is red. And so we have that in each of these, and so that way we have, we can tell if this is GMO positive or not. So yeah, that was jazzy. Okay, so after we put all of those tubes that I just talked about in the thermocycler, after that's all done, we start them in the PCR. And that's gonna take 3 to 4 hours to complete, and so that was fun. I'm sure, Ms. O'Hara, you had fun with that. So then we gotta store our samples in the refrigerator until the next day. I think that the next day we did the next part. And then we move on to part 3, which is just running the gel, which we've done before. Okay, so now we get to talk about our results. So after we ran the gel, none of our samples really showed up that well. So basically we had a very clear GMO positive with PR and with PM and GMO positive positive with GM. So that was fun. So basically we didn't really get the results we hoped to, because they didn't really show up. But we would know. Let's say my bean was GMO positive, and so let's say this is my bean. And so if it is GMO positive, then it would be right with this one. And if it was GMO negative, then it would be with this one, because these are our controls. And then our GMO negative control would be here. And so that's how we would know. But it didn't work out that way, so that was fun. But yeah, our results were kind of inconclusive. We don't really know if my control was GMO. I mean, my test food was GMO or not. So yeah, that's the whole shebang.